Hello and welcome to my presentation about effects of spaced k-mers on alignment-free genotyping. My name is Hartmut Häntze and I'm a research associate at Charité University Hospital Berlin. Previously, I've been a graduate student at the National Chong Kung University in Taiwan, where I wrote this article together with my advisor Paul Horton. To start with, I would like to show you a small headline from last year. Illumina reveals new high-throughput instrument NovaSeq X. With this system, one can sequence a whole human genome with 30 times genome coverage. That's around 120 gigabits of data per hour. This is great news for many fields such as population studies, where we need to sequence thousands of genomes. But obviously, now we don't only need software that is accurate, but we also need software that is very fast. Otherwise, we will never terminate. So let's have a quick glance at possible candidates that could handle this. With this, I mean genotyping, that is the process of determining which genetic variant an individual possesses. There are many groups of algorithms that are alignment-based, so they take the reads and then they first map them to a reference sequence and afterwards use this to call the variants. This works very well, but it is also quite slow. Depending on the data you have and the hardware, it can take from hours to even days. Now, another group of algorithms is the so-called alignment-free group, like LABA, BiSTable, or PanGene. They completely skip the mapping from the reads to the reference sequence, and instead they have a database of already known variants, and then they directly map the variants to the reads. This accelerates the problem. Now, we use both algorithms in our lab, and we realized that the algorithms on the left hand, BWA or Bowtie, they often use a seed and extend strategy, where they have a spaced seed and then based on that extend it. And this first of all accelerates things, and second of all, it makes them more resilient to mutations or sequencing errors. So now the algorithms on the right side, they don't use that, maybe with the exception of LABA. And we found it a little bit weird because, right, the spaced seeds might not be helpful when it comes to accelerations in the right case, but they should show the potential to make them more resilient to sequencing errors. Which is now the contribution of our paper. We try to answer the question, can spaced k-mers improve k-mer-based alignment-free genotyping methods? To answer this question, we used already existing software, PanGene, and we added a spaced seeds functionality. This is great because now we can use this software PanGene with both contiguous and spaced seed and compare the performance of both kind of k-mers. Our application is publicly available on GitHub. But let me first introduce you the foundation of our work. One interesting article is fast genotyping of known SNPs through approximate k-mer matching. They introduce a software called LABA, which has a similar idea to our project. They want to reduce the impact of sequencing errors by implementing approximate k-mer matching, which allows matches up to a Hamming distance of 1. So if I have a k-mer of length 31 and there is a single sequencing error, then it can still be matched to a reference. They show that it indeed reduces effects of sequencing errors. However, one disadvantage is that it also decreases the number of identifiable unambiguous loci in the genome. So if there are two regions in this reference sequence which are very similar and only have a single base as a difference, then it's really hard to distinguish both of them. The paper that had the biggest impact on our work is pangenome-based genome inference allows efficient and accurate genotyping across a wide spectrum of variant classes by Jana Ebler et al. And this is again a k-mer-based genotyping algorithm. And one of the biggest contributions of this paper is that they switched from a reference sequence to a reference pangenome, which first of all reduces reference bias and also allows the algorithm to do haplotype-based genotyping. Now, we wanted to take this software as a foundation and now replace every step where they use a k-mer with our spaced k-mers. But first of all, I need to talk about spaced seeds, which are the blueprints for spaced k-mers. They were introduced to bioinformatics in a very influential paper by Martrom and Lee, where they showed that they can indeed increase sensitivity of matching algorithms. In their case, it was homology search. A spaced seed is simply spoken a mask of 1 and 0, and we can shift this mask about on a sequence and then iteratively just take the basis below a match position, which then would create our spaced k-mer. Let me show you on a small example why spaced seeds can help to increase sensitivity of matching algorithms. Let's say we have a reference and a read, and the read clearly belongs to the reference, but unfortunately there is a sequencing error right in the middle of the read. Now on the left page, let's say we have a contiguous formula and we want to look for matching bases, but there is not a single complete match between the read and the reference, so we cannot associate the read to the reference. However, if we now add a don't care position in the middle of the seed and transform it from a contiguous to a spaced seed on the right side, then we are able to find a matching positions between read and reference and can now say the read belongs to the reference. So let's have a look at our problem setting. In the PanGenie paper by Ebler et al., they use standard k-mers with a length of 31. So this is going to be our control group. We also designed a seed of length 31 and weight 31. Now we want to compare this seed against two spaced seeds, which you can see here. They do have the same length of the same weight of 31, so there are 31 ones and then additionally 20 zeros. We designed both of these seeds with the help of the software called Speed. Speed is a helpful tool which allows us to fast compute good seeds. Now we know that Speed is not actually developed for this kind of context, but the problem is NP hard. So while Speed is not the perfect solution, we still think it is a good solution for this kind of problem, which is also similar to the problem Speed was developed for. We're going to compare both of these seeds to the control group. And we want to see the impact of these zeros, the don't care positions. Now, obviously, there's actually a second difference between the control group seed and the spaced seeds. That is that the control group has a much smaller, much shorter seed than the spaced seeds. So possible differences between spaced and control group might also be caused by the size, by the length of the seeds. So to guarantee that this is not the case in our final results, we added a second control group, we call it C prime, which has the same length as the spaced seeds. So now if we see a difference between the spaced seeds and the control group C and C prime, we can be sure that it's only due to the don't care positions. We evaluate our program on reads from 9 individuals from the NCBI sequence read archive. And we do have the reads in 5 and 30-fold sequencing depth. They are all short NGS data with 150 base pairs length. Our ground truth is going to be haplotype resolved variant call files for all of these 9 individuals, which origins from these two papers I mentioned here. Let's have a look on our results. These bar plots show the weighted genotype concordance of all 9 samples having 5-fold coverage for single nucleotide variants, indels, and structural variations. Genotype concordance is the proportion of correctly called variants. And it is weighted because we do have variants that are not in the genome of the sample at all. So there are homozygous reference. Some of them are heterozygous and some of them are on both alleles, so they are homozygous alternate. Now, the first two bar plots are always the genotype concordance of the control group, and the last two bar plots show the genotype concordance of the group with the spaced k-mers. And as you can see, the spaced k-mers outperform the control group in all cases, independently of variant type. Now, if we look at the data at 30-fold coverage, the results get a little bit more complex. First of all, the results are obviously better because it's more coverage, but now we also see that for the single nucleotide variant, the spaced seeds perform as good as the control group, so there is no difference. However, in the indel and the structural variant case, again, the spaced seeds are better than the control group and the results are significant. Let's have a look at absolute numbers. These are the results for arbitrarily chosen sample NA12878, a woman from Utah. So in our database, we do have around 13 million variants. And we try to genotype these variants in this sample, having 5-fold coverage. Using the control group contiguous seeds, we achieved around 11,500,000 correct variants, which is already around 89%. Now, after we use space k-mers based on seed 1, we could improve the result up to 11,800,000 correctly called variants, which is 300,000 variants more. Having 30 times read coverage, it gets a little bit more complicated. We see that the spaced k-mers now perform as good as the contiguous k-mers. Those have a proportion of 97%. And if we look at the absolute numbers, we see actually the spaced seeds performs even a little bit worse. Now, an interesting side effect which we observed in our experiments is that not only perform the spaced seeds exceptionally well, but also the long contiguous seeds. In fact, in all cases, the long contiguous seed C prime performed better than the short contiguous seed C. And we had a look at the variant call files and tried to understand why that is. And it appears that in many cases, the short seed did not find any unique k-mers that can uniquely associate it to a reference region, while the long k-mers and also the spaced k-mers, which are long as well, could often find unique k-mers and therefore performed better. However, the seed length alone cannot sufficiently explain the increased performance because the spaced seeds still performed better than the contiguous seeds in many cases. So we conclude that the improvements are very much likely because of the don't care positions in the spaced seeds, as they make the spaced k-mers more resilient to sequencing errors and help the algorithm to improve the results. For future research in this topic, it would be interesting to see whether different seeds also show different genotyping results. Further, it is necessary to decrease the runtime of our algorithm's mask-pangeny, because at the moment it is somewhat slow due to its rather naive implementation, so we are still lacking multi-threading and so on. However, I'm confident that there are going to be some updates soon, because I know that my previous lab mates are working on this, and I hope that they can, and they already have results, and I hope that they can publish something soon. Last but not least, it would be interesting to evaluate the algorithms on also data sets from different sequencing platforms to see in what kind of aspect they can generalize. To sum it up, in our article, we proposed an algorithm that utilizes spaced k-mers for alignment-free genotyping, and we believe we are the first ones that did that with exact k-mer matching. Our spaced k-mers could increase recall, F1 score, and weighted genotype concordance of genotyping. The effects are particularly large on low coverage data, so we recommend using spaced k-mers, especially on this kind of data. And we believe they can be a valuable technique for improving alignment-free genotyping. Thanks for your attention.